Hello everyone, and welcome back to Upregulate, the channel where we discuss everything stem cells, gene therapies, and the future of advanced treatments. Red blood cell concentrates are widely used for treating patients with blood loss or anemia, and there is increasing demand observed everywhere in the world. Blood donations are not always sufficient in accommodating all those in need. Furthermore, blood group donations require blood group and antigen identification for matching with the recipient, and viral safety must closely be monitored. These elements of the blood donation supply chain can be costly, time-consuming, and convoluted if supply chains and organisations are not managed sufficiently. In October 2022, NHS Blood and Transplant triggered an Amber Alert on blood stocks, meaning hospitals have been encouraged to put in place management plans to protect their blood stock, a move which could mean postponing some non-urgent elective surgeries to ensure blood is prioritised for patients with urgent blood requirements. Blood can only be stored for 35 days, meaning there is a constant need for donations, and a need for specific blood types. Culture or manufacture of red blood cells at industrial levels aim to make red blood cell concentrates available at large scale anytime and anywhere in the world. Developing, optimizing and scaling up red blood cell culture at a controlled and economically sustainable cost is therefore a primary objective for improving and standardizing red blood cell availability for all blood groups. Which is excellent news for all my fellow vampires out there. Technical reports have shown that mature, fully functional red blood cells can be produced from various types of source cells, with low level of contamination and the potential to be used in transfusion therapy. Studies have further showed that optimization and cost reduction of the red blood cell culture process has not yet been reached. Could red cell manufacturing be a future solution to low blood stocks and donations across the globe? Blood cell culture requires multiple factors or conditions to be controlled to ensure suitable yield and viability. An initial stem cell source is required for the beginning of production, with suitable conditions to obtain mature and fully functional red cells. Multipotent progenitors harvested from umbilical cords and the peripheral blood have been used in production. CD34 positive stem cells are selected for initiating this red blood cell production in vitro. However, starting stem cell sources must be the closest to the final produced cell. This is to shorten the culture time and cell availability. This has been achieved in laboratory conditions for platelets production, which can be cultured within a few hours from megakaryocyte progenitors. However, red blood cells require a much longer time to culture. The culture of mature and functional erythrocytes spreads over 3 weeks and requires multiple mediums and growth factors, which cause the cost of production to skyrocket. The major challenge in upscaling this red blood cell culture process is controlling the required amounts of high-cost growth factors, bioproducts such as erythropoietin and insulin, culture medium and additives in production, which render the bioprocess very expensive. Unfortunately, due to the difficulty in affordable upscaling of this process, in vitro produced erythrocytes still remain restricted to laboratory use. The transfusion objective appears not yet reachable within a short time, as challenges for the large-scale production in bioreactors at an affordable cost are not yet foreseeably met at practical levels. This field remains at this time an active research field, waiting for a breakthrough for improving the production, transferring it with optimized growth conditions to large bioreactors, and shortening the total time required for obtaining mature and functional erythrocytes. The RESTORE trial is a joint research initiative by NHS Blood and Transplant and the University of Bristol to assess the recovery and survival of a mini dose of red cells derived from CD34 positive cells isolated from adult blood versus the standard donated red blood cells. Basically, cells which are cultured in a laboratory will be tested against standard donated red blood cells. This study hopes to investigate the lifespan of the grown cells compared to the infusions of standard red blood cells from the same donor. The hypothesis of the study states that the manufactured red blood cells will have a longer survival in circulation of the volunteers than the standard donated red blood cells. The trial team expect that laboratory-grown cells will perform better than standard donated red cells, which contain cells of varying ages. The success of this study could benefit the small number of patients with rare blood group types and diseases, for example, thalassemias and sickle cell disease, that NHS Blood and Transplant cannot meet the transfusion requirements for. It can be challenging to acquire a suitable amount of donated blood for those struggling with certain medical conditions. It is possible that by culturing certain red blood cells from human blood cells in a lab setting, a novel form of transfusion could be offered to those in need, including those requiring blood transfusions for the duration of their entire life. Moreover, if it's found that cultured red cells can sustain for a longer period in the body, those who require regular blood transfusions may not have to receive them as often. This would decrease the accumulation of iron in the body from constant blood transfusions, which can cause severe health issues. As of November 2022, the trial is reported to have transfused 2 research volunteers with between 5 to 10 mils of cultured red blood cells. This is the first time in the world that manufactured red blood cells have been given to another person as part of a blood transfusion trial. Further investigations are essential prior to implementing this in clinical practice. Yet this investigation denotes a noteworthy advance in utilising laboratory manufactured red blood cells to enhance the care of patients experiencing uncommon blood types or individuals with intricate transfusion needs. With recent amber alerts on blood stocks in England, in the future this trial could hold the answer to the low blood stocks and donations. For now, NHSBT continues to rely on the generosity of donors. Blood transfusion is an essential and irreplaceable part of modern medicine. Despite the necessity of blood donation in modern healthcare provision, the donation and identification process of red blood cells requires significant resources to maintain. The scarcity of some donors, potential transmission of pathogenic microorganisms, and overall costs of processing of each unit have switched scientific interest to the quest of alternative transfusion methods. The industrial manufacture of transfusible red blood cells looks to be a more appealing prospect year on year, as maintaining suitable blood stocks for a growing population becomes more difficult, and studies such as RESTORE are attempting to make this idea possible. By further studying and refining the process of stem cell manufacture, it is expected that the financial and scientific dilemmas of the current approaches will be resolved in the foreseeable future. Hi everyone, thank you for watching today. If you're interested in the papers that I researched for this video, then I'll be sure to link them at the end of the video and in the description as usual. If you're interested in the RESTORE trial, I'll also add a few links to do with that. That research is amazing, and as I've described in this video, it could be the future of blood transfusions. As I say, thank you for watching today, and I'll see you in the next video.